Hello everyone, today I'll be presenting my research that I've done at RIU, NJIT. My name's Nicolette Filippone and my major is environmental science. I currently attend Bergen Community College. On the top left-hand side of the slide is the DNA that presents the database I will be collecting from the cancer patients at NIH, which then is computerized with Linux machines and commands for the exome sequencing on the top left bottom that goes into the final step, which is to find the variants associated with cancer. So my goal is to determine the variants associated with colorectal cancer through exome sequencing. Exome sequencing consists of two steps. First, selecting only the subset DNA that encodes proteins, which are the regions known as exons, and the second is the sequence exonic DNA using high throughput DNA sequencing technology. Here's a little diagram of how the method works. So the problem statement, as all of you know, cancer is a huge dilemma today that is a field in between biology and medicine. Cancer risk prediction is a problem of great interest today also. My project in the future would go towards a platform that could predict someone's cancer risk depending on their gene expressed or alleles. Now, how this work in the future consists of having your DNA put through a website, which then calculates the different genes and alleles that have the percentage of the disease risk or cancer risk. What my project concentrates is just finding the variants associated with cancer using Linux machines and commands. I just thought I would say that to give the overall picture. The mutations that I'm currently working with is somatic mutations, which occur in the non-germline tissues, and it's not heritable. So a little more information about colorectal or colon cancer is that it is the second leading cause of cancer in the United States. 90% of new cases occur in people 50 or older. People with a first-degree relative parent, sibling, or offspring who has colon cancer have 2 to 3 times the risk of developing the disease. There are currently more than 1 million colon cancer survivors in the US. Now you may ask, what if we did have this platform? Would it be open to the public? And the answer is, from speaking to my mentor, Professor Usman, it would be, which would be a great development for now and the future, so that people can find what environmental factors, what styles they should do in order to stray away from cancer and cancer risk. The significance of this research is that, as what I said, we can make a platform for people to predict. We can suggest certain drugs, people can neutralize the predictions. And in this slide, background and approach is the process that I will be dealing with. Now, on the top left with raw reads, which is the pre-processing, I would get the NIH database with the colorectal cancer, map it to the human reference genome, do a little bit more work, mark duplicates, base recombinate, and then for the variant discovery, I will use a software program, something called MuTech, and then filter the variants and then have the variants at the end. Now you may think finding the variants associated with colorectal cancer is very easy. It could be easy for a computer genius who knows Linux machines and Linux commands, but it wasn't easy for me due to my lack of knowledge. This RU past summer research, I've experienced immensely in Python and Linux machines and Linux commands, learning what organization, what indents, and what positioning these letters, characters should be. So for me, it was a huge success and a huge progress. Experimental design and methods. So as I said, we compare the genomes in order to find the mutations. We use the Linux machines and Linux commands. We use Python to program as a pipeline to interface all the programs together. The programs we used was Cosmic, Mutek, Sambamba, and a few others. And in total, we analyzed a subset of 2.5 terabyte data of exome sequencing. In this diagram, finding the variants, here is a little picture describing what the machine learning vector would be doing. As you can see here, this line represents the human genome reference. Each vector displays a different scenario. The first one would be the mutation, and as you can tell, the A and G do not match, which represents a homozygous SNP encoded as 2. Over here, 2. In the second one, you can see that it has 2 different alleles, which is a heterozygous SNP encoded as 1. Here, all of them are the same, which is why it represents 0 for no mutation. And then the last, it is missing, heterozygous indel encoded as 1. In the future machine working, these would be the numbers representing into a future vector of 4 dimensions: 2, 1, 0, and 1. And that is how the machine would be finding the variants. So my results, I have gone up to here, which is the SNV and indel calling. My next step, which I am finishing up, would be the last one, which is to obtain and receive the variants that are associated with cancer. This process has taken me long. Each time you put the process in the command, it can take up to 6 to 12 hours depending on how big your data set is. Future work. So we need the alignment of the short reads, which is aligning the DNA to the human reference genome of the cancer patients in order to determine correlated mutations, which will be used in machine learning context to predict a cancer risk platform. The machine learning is done by converting the mutations into future vectors, which is shown in the finding the variant slide. And if time permits, we will study the support vector machines, random forests, and neural networks for predicting colorectal case and control studies as the final step. Thank you.